Okay, so you received two videos for meiosis 1 and meiosis part 1 and meiosis part 2. That should be watched in the beginning part of your break week. I mean, you could do all of this at once. That seems unwise. But so we're going to do that information, protein synthesis, and if we can fit it in, one more thing during break. My goal is to make these really short for you so that it's not a ton of work over break, but if you are really dedicated about and you really want to get that for that 5, watching these is going to be so important because we are not going to take a ton of time to do it once we get back. So we've talked about DNA replication, mitosis, meiosis. We're now talking about how do I make proteins from that DNA. Proteins, one of our 4 macromolecules, are what we call the workhorse molecule. They are basically the things that are found in all organisms, and DNA is the blueprint that allows us to code for proteins. Proteins carry gases like hemoglobin, they are enzymes, they provide for structure, they are antibodies, they do movement, they store. It's just a lot of different things that they do, and we can't function without them. The history of this, which not super important, jot it down, but don't memorize it. Archibald Girard suggested that genes determined phenotype through defective enzymes controlling biochemical pathways. What that means is that he basically thought that the way that we physically looked was determined by enzymes and their control, and he wasn't that far off. Beadle and Tatum established that genes basically created our enzymes, and they studied bread mold. All of this basically allows us to arrive at these conclusions. One gene produces an enzyme, and we find out later other proteins. And so they modified that finding to say one gene always produces one protein, and then modified it a little more to say one gene actually produced one polypeptide chain. But the idea is that we have tons and tons of genes. They're the little bands on our chromosomes, and they code for certain proteins. Anything from whether the lactose enzyme, so it'll determine if you're lactose intolerant or not, to the color of your hair, how quickly your nails grow, stuff like that. The overview is DNA is located in the nucleus and proteins are made in the cytoplasm. And so RNA is the intermediate between DNA and RNA. What happens is in the nucleus, DNA is transcribed to make RNA, and then in the cytoplasm, RNA is translated to make proteins. The structure. The most important part, right, is that in DNA we have our deoxyribose, we have our phosphate, and we have our purine or our pyrimidine, so A or G and T and C. The main difference in RNA is that we have ribose, so different sugar, and we include uracil instead of T. We still include A, G, C, and just include U as well. There are 3 main types of RNA. mRNA is the RNA that is actually read to create a protein. tRNA is going to come up later. We'll talk about that a little bit more. And then rRNA creates a functional ribosome. We'll talk a little bit more about these. In prokaryotes versus eukaryotes, prokaryotes don't have introns like eukaryotes do. We'll talk about what an intron is. RNA in prokaryotes does not have to be processed like eukaryotes, and transcription and translation happen at the same time in prokaryotes. It's also because they don't have a nucleus, so the process isn't split up. It's just a simpler process in general is probably what we should know. Amino acids are coded for by a triplet of DNA nucleotides called a codon. We've talked about this, right? That my long string of DNA letters, every 3 codes for a certain amino acid. So if we look, we see my DNA strand has ACC, AAA, CCG. I transcribe to make my strand of RNA, and then UGG codes for a TRP or amino acid. UUU codes for an FEE amino acid, and each of those is like a little amino acid in my protein chain. Oops. These guys were the first to discover that that a codon, a set of 3, codes for an amino acid. I wouldn't really worry about this. The code has redundancy. Basically, that means that there are chunks of letters that code for the same protein. If you look at the diagram, let's say for SER, UCU, UCC, UCA, UCG, they all code for the same amino acid. This code is also universal, which means that almost all cells use it, and eukaryotes as well as prokaryotes use this as well. So when we're creating a polypeptide, what we look for is a start and a stop codon. If we look back, we'll see AUG is my start codon, and there's a couple different varieties that can be a stop codon. So we're going to start from beginning to end, talking about what happens. This is transcription first, so it's happening in the nucleus. It's the RNA synthesis from our DNA, and there are 4 steps. Okay. Excuse me. Initiation. Initiation is going to be our first step. It means that I find what we call a promoter, which is our starting point, and it has what we call a Tata box. The Tata box is basically a series of nucleotides that signals this is where you start making RNA. So we have our promoter. We have basically transcription factor, which is an enzyme that's going to link up to my DNA, specifically around the Tata box, which is a series of letters. And RNA polymerase 2 is going to envelop this and start transcribing the DNA into RNA. Elongation, RNA polymerase unwinds the DNA and base pairs RNA to the DNA gene. So elongation is the physical creation of my RNA strand. Termination is the physical end of creating my RNA strand. And then RNA processing, basically what we have to do is put the finishing touches on. So we put a cap on the 5 ends. They add about 30 to 200 adenine nucleotides to create what we call a poly A tail, and any introns are removed, which is like sequences of material that we don't need. The 5 cap protects the RNA and keeps it at a certain length. Same with the poly A tail, similar to telomeres in DNA. Introns, these are going to be sequences of codons that are not useful. And so we use spliceosome to remove the introns. Spliceosomes are composed of smaller particles called SNRNP. They're proteins. Basically, the spliceosome will splice the intron at the specific start point and cut out that useless part of RNA and then fuse the two together. The exons are what are going to physically code. The introns we get rid of them. So in RNA processing, which is the last step, different exons are recombined in different ways for certain mRNA. This increases the number of proteins that I can make. So I can leave some introns in if I need to. It will make the protein I make different than if I splice out certain introns as well. If we look up there, we see that I have the pre-mRNA, and during processing on the left, I've taken out the green. On the right, I've taken out the yellow, and 2 different proteins are formed. So we call that exon shuffling. Proteins often have to have certain regions we call domains that allow them to bind to other proteins. Think about like hemoglobin is actually 4 proteins stuck together. In a lot of cases, exons code for the different domains, and so depending on where I put the exon, which ones I cut out, this can create certain new proteins or create one protein versus the other. At this point, I think my video is frozen. I don't know if this is still going, but at this point, the mRNA has been processed, and it's called a mature mRNA, and it's ready to go to the cytoplasm for translation. And that is the end of the first part of this.